Hi students, hope you guys are fine today. So we are moving on to the part of 2 fundamental unit of life biology of class 9. So without wasting time, let's hop on to the chapter right now. We are going to start with the part of nucleus. So nucleus is a darkly colored spherical or oval dot-like structure near the center of each cell. So explaining the structure of nucleus is nucleus has a double-layered covering called as nuclear membrane. The nucleus is filled with a fluid called as nuclear plasm. The nucleus has a membrane which is porous in nature, which helps in transfer of materials inside and outside of the nucleus. It also has a cytoplasm. The nucleus has chromosomes, chromatin material, DNA, gene, and many more important items constituting in it. So let's move on with chromosomes right now. Chromosomes are visible as rod-like structures only when the cell is about to divide. The chromosomes contain the information for the inheritance of characteristics from parents to the allele which means the offspring or the next generation. The DNA stands for deoxyribonucleic acid, and each chromosomes are constitutive or composed of DNA and proteins. DNA is an important component and an essential part of our daily life as we are made in the alignment of protein only by the DNA's transferring inheritance details, or in simple words we can say that DNA has a gene in it. Gene is the element that has been transferred or inherited from our parents, thereby we have similar characteristics to our parents, like the same eye color, hair color, and facial structures, characteristics, habits, etc. These DNA are presented in the chromatin material. Chromatin material is a visible entangled mass of thread-like structures. Whenever the cell is about to divide, the chromatin material gets organized into chromosomes. The nucleus plays a central role in reproduction, process by which a single cell divides to form new two cells, and it also helps in determining the way by which the cell will develop and what form it will exhibit at maturity by directing the chemical activities that has been taking place inside the cell. Now we are going to see the two types of cell that is based on the classification of the nucleus type, which is prokaryotic and eukaryotic. Prokaryotic cells are generally small in number. There would be nearly 1 to 10 yum. 1 yum is nearly 10 to the power of 6 meters. Second, the nuclear region of prokaryotic cell is not well defined. It does not have a nuclear membrane. It is called as a nucleoid. A single chromosome is present in a prokaryotic cell, and the membrane bounded cell organelles are absent in prokaryotic one. Whereas now let's see the eukaryotic one. Eukaryotic are generally large in size when compared to the prokaryotic one. There would be 5 to 100 yum, and 1 yum is 10 to the power of 6 meters, and thereby when we classify them on the basis of the nuclear region, eukaryotic cells are well defined. They are surrounded by a nuclear membrane. Third, on the basis of chromosomes, they have more than one chromosomes in eukaryotic cell. Okay. And fourth one, we are classifying on the basis of the membrane present in the cell organelle, which is present in eukaryotic cell. Now let's see about the cytoplasm. Cytoplasm is a region where in a cell which has a lot of fluid in it and in which all the cell organelles are suspended. These fluid are rich in proteins and many other fibers that enrich each cell organelle and provide them with necessary nutrients to perform their activities, cellular activities. Thereby, now let's move on with cell organelles. What are cell organelles? They are large complex cells including cells from the multicellular organism. They need a lot of chemical activities to support their complicated structure and function. To keep these activities of different kinds separated from each other, each cell has a membrane bound little structures called as ganels within themselves. So, these functions are performed by each of the cell organelle of its specific functions. So, moving with the most important cell organelle, which is endoplasmic reticulum. Endoplasmic reticulum is also denoted by ER. Endoplasmic reticulum is a network of membrane-bound tubes or sheets. It may look like a tubular structure, round, oblongated bags or vesicles. ER is a similar structure to plasma membrane. ER are two types, which is RER, SER. RER stands for rough endoplasmic reticulum, whereas SER stands for smooth endoplasmic reticulum. Rough endoplasmic reticulum looks rough under the microscope because it has ribosomes attached to its surface. And these ribosomes helps in the protein manufacturing. So, in ER is the main hub of protein manufacture in a cell. Whereas when compared to SER, they do not have manufacturers of protein or ribosomes in them. So they are smooth in structure when it is being observed through a microscope. Then they have a lot of fat molecules or lipids in it. And these proteins and lipids help in building the cell membrane. And this process by which a cell membrane is made using the lipids and proteins generated by the fat molecules present in SER is called as membrane biogenesis. These lipid functions as enzymes and hormones inside the cell. They help in forming or performing the functions through a network system. One function of ER is to serve as channels for the transport of materials from various regions of the constitutive cell. So moving on with Golgi apparatus. Golgi apparatus is first described or discovered by Camillo Golgi. Camillo Golgi is a great scientist born in Curchinio near Brescia in 1848. He studied in the Medicine University of Pavia. After graduating in 1865, he worked at Pavia at the hospital of St. Mattico. So, moving with Golgi apparatus, it is a membrane-rounded vesicle which are a furthermore expansion of ER. ER tubules are consecuted in a curvier structure to make Golgi apparatus. Golgi apparatus have cisterns that are arranged parallel to each other, and these membranes are connected to form a cellular membrane structure, helping in digestion as well as furthermore transportation of the materials that are made inside the cell. They dispatch various materials inside and outside the cell through Golgi apparatus. So the main function of Golgi apparatus is storage, modification and packing of products in vesicles. They also convert complex sugar into simpler sugars, and it also helps in the formation of lysosomes. So moving with the lysosomes right now. So lysosomes are also membrane bound cell organelle that has digestive enzymes in it. These digestive enzymes are made by RER, which is rough endoplasmic reticulum, and they perform an activity of waste disposal system in a cell, and they keep the cell clean by digesting the foreign materials that enter through our cell using the plasma membrane, as it is semi-permeable, and they are worn out cell organelle too. They kill the bacteria and the harmful food materials that enters through our cells. They are a component that has to break the complex substances into simpler one. They are able to digest even the um, powerful materials using their digestive enzymes. So thereby, during the disturbance in the cellular metabolic activities, they might be damaging their own cell, and they might burst out releasing their enzymes digesting their own cell. So they are also called as suicidal bags. So now moving with mitochondria. Mitochondria are the powerhouse of the cell. They have two membrane coverings. The outermost covering is porous, whereas the innermost covering is deeply folded. So the innermost folds are quite important as they increase the surface area for the ATP generation, and ATP are the energy currency of the cell that helps in providing energy to the cell organelles to perform their basic structures and functions. These are released by mitochondria, and ATP has a full form of adenosine triphosphate. The body stores energy in the form of ATP, and they help in forming new chemical compounds for mechanical work too. And these types of organelles might have this strange quality to make their own DNA, and they also have ribosomes in it. So, mitochondria can also make their own protein. Now, let's hop on with plastids. Plastids are present in only plant cell. They are chromoplast, leucoplast on the basis of classification. Chromoplasts are colored plastids, whereas leucoplasts are white or colorless plastids. Chromoplasts contain pigments like chlorophyll for its green color, and chloroplasts are important for photosynthesis in plants, whereas leucoplasts are white in color. They have starch, oil, and protein content in it. They help in storage of materials, as well as these types of cell organelles have numerous membrane layers embedded in the form of stroma. They have parts like grana, stroma in it. Plastids are the second type of cell organelle that has their own DNA and ribosomes. So plastids also have a function of making their own proteins using the ribosomes that are present inside it. Now at last we are going to see the last cell organelle, which is vacuoles. Vacuoles are a sort of storage sacs in solid or liquid content. They constitute 50 to 90% of the cell volume in plants. Plants require a lot of energy needed to be stored so that they can be stationary, as well as they can help the cell to stand upright. Vacuoles are small in size in animals, whereas large in plants, and they are situated in the center of the cell. Vacuoles helps in providing turgidity and rigidity to the cell. Plants stores amino acid, sugars, and various organic acids and some proteins in vacuoles. Amoeba also forms vacuole using their pseudopodia of false feet for secreting the digestive enzymes for digesting their materials or the food particles that are ingested using the false feed or the pseudopodia from its external environment. Thereby, at last we are going to see the cell division. Cell division is a process by which new cells are made from the older one. There are two types of cell division. The process of cell division, in which most of the cells divide for growth purpose, is called as mitosis. In this process, the mother cell divides to form two identical daughter cells, and the daughter cells have the same number of chromosomes too. So they help in growth and repair of tissues. Now